Good morning, everybody. Morning. You just came from a business meeting, so you're like, yeah. Or is it just that early for you all? I'm still waking up at like 3 in the morning myself, so I've been up and had 10 coffees already, so I'm fine. But it's really great, again, to be here. I've just enjoyed myself thoroughly. Can't thank the organizers enough for thinking of me and bringing me and my daughter all the way down here for the last week or so. Us Yanks can also learn a lot about the organization of the meeting. Usually by this time, we're about an hour into the meeting. We're about an hour late already. So, you guys are run on a Swiss watch as far as I'm concerned. So this is really great. Thank you again, and I've heard all wonderful things, and they're all true, and then some. So I'm not going to be talking about Queens or really any of our research at all. This was a topic that was selected and requested because apparently, even though it's June, it's winter for you, right? Up in the Northern Hemisphere, we're about to enter into that unbearable season of summer when nature just completely shuts down, the bees get all robby, there's really no kind of nectar flow coming in and everything, and it's just a very difficult time of year. And then, of course, the mites start getting out of hand, and, spring is when beekeeping is really easy and fun, and it's all downhill sledding, and we're about to enter in that phase where it's kind of uphill sledding. But for you, it's the winter, so, I think this is a timely topic. And really talking about the biology of overwintering and what we as beekeepers can do to kind of come out of winter on a springboard and really be off and running. So this might be kind of introductory for a lot of you. Hopefully not. Hopefully there'll still be some kind of tidbits in here. But just kind of talking a little bit about the biology of overwintering honeybees. And it's my contention, again, I'm biased because I'm a biology professor after all, but my contention is that beekeepers really benefit by understanding the biology of honeybees. And this is something that, at least in North Carolina, if not the US in general, we don't do as much of a good job at the beginning levels of our bee schools and our beekeeping short courses. They're often talking about how to, not the why. So understanding the biology of the bees is really to understand the why. And I like to tell beekeepers in my neck of the woods that I actually never read a beekeeping book until about the 6th year that I was keeping bees, because I understood the biology of the bees, and all the rest of it is just putting boxes on top and smoking them every once in a while. So if you really understand kind of the motivation and the basic biology of the bees, that really improves our beekeeping. And it's the difference really between cooking by following a recipe versus being a master chef, and you don't need to measure everything, you're just kind of winging it, right? Anyway, so talking about the coming out of winter, this is really what I'm going to be talking about, at least the colony losses in the US, just very briefly talking about that and how overwintering, mortality is a major concern as it is here. And Pike's presentation yesterday actually had a lot of really interesting and fascinating parallels with what we have up in the US. Then we'll talk about kind of going into winter, because coming out of winter really relies so much on what we do going into winter. And then we'll talk about just kind of spring management in the early, middle, and late stages of that spring management, ending with swarm management. So as was alluded to by Peter and by Pike and others, there are a lot of colony losses in the US. And over the last decade or so, the average has been in the US has been about 40%. Now again, this is a turnover rate, not a constant decline. There's a turnover rate of about 40%, so every winter and every season, beekeepers in the US lose about 40% of their colonies, and then they have to grow them back. So there's this kind of unsustainable oscillation of the managed honeybee population that this blue bar right here, averaging around 15%, is the acceptable loss, as was talked about yesterday in your surveys. So again, it exceeds that, so these are unsustainable rates of losses, annual losses, where we have this kind of fluctuating population that makes it very tenuous for the horticulture and other sectors in agriculture that rely on pollination. Now again, as part of our surveys, or the Bee-Informed Partnership surveys, they're asking the beekeepers, why do you think your bees died? Now again, this is not kind of scientifically ground truths in the same way, and there can be multiple reasons, so these columns don't add up to 100%. But the top reasons are, kind of starvation or a colder than normal winter, they're weak in the fall. All of these things are kind of predicated on going into winter properly so that you can come out of winter. So if you're not doing what you need to do in the fall, then you'll have winter losses. It's just kind of that simple. But the numbers, again, are quite astounding. I'm just flabbergasted that you guys are complaining about a 13% annual loss. Wah. So 40% is what we've been dealing with for quite a long time. Of course, there's all sorts of reasons for that. We have these kind of large-scale mechanized things, operations. 90% of our beehives are owned by 10% of the beekeepers, the largest of whom I think has 100,000 colonies. So, it's just kind of at a different scale. Anyway, so let's talk a little bit about kind of the winter biology and the winter preparation that we need to do as beekeepers going into winter. And please, again, if there's anything that this doesn't apply in the southern hemisphere, just raise your hand and tell me to move on. But this is what I tell a lot of our beekeepers in North Carolina. And I really start with these three famous adages. Well, two of them are famous. One of them I just made up and say it's famous. The first is taking your winter losses in the fall. This hopefully is a phrase or at least a practice that you guys do here as well. It is far, far better to take two struggling colonies or kind of borderline colonies and unite them using the newspaper method or whatever other methods that you use here, and have one strong, well-fed colony going through the winter, that the success rate is going to be much higher for that one colony, and then later next spring you can split it again. But trying to nurse two borderline colonies through the winter, you're usually going to end up with two dead colonies. So this famous adage of taking your winter losses in the fall is very, very appropriate. The second is that bees don't freeze to death, they starve to death. From what I can understand here in the northern island at least, it's very similar, at least the same parallel south, that North Carolina is north, about the 35th or so parallel. So the winters are quite mild, I think, compared to, in our case, up north, in your case, further down south. So depending on where you are in New Zealand, the winters can be, to a greater or lesser degree, more severe. But in general, as long as the bees have food and as long as the bees are healthy going into winter, they'll continue to generate the heat that is needed waiting till next spring. And so, you need to make sure then that you have sufficient honey stores, and you want to make sure that it's above the cluster, and we'll talk a little bit more about this. And then finally, the last famous quote that I just made up is that these winter bees, right? I think there's a greater recognition now that these diatomists or winter bees are really, really crucial for overwintering, which again is obvious once you think about it, but I don't think in our beekeeping practices, at least up in the US, they've really been thinking about that, especially down south where we are, where they don't have as long a winter. But I think actually that it might become more important actually the further south you are, where there doesn't seem to be an overt winter outside, but it's actually really crucial inside. And so what that means is the bees as a colony are raising these special long-lived winter bees we need to make sure that they are as healthy during development as possible, which of course, this is the same time of year, the same time of season, when the varroa mites are really ramping up. So we really need to make sure that we're keeping those varroa under control so that those winter bees can be raised without being weakened and therefore compromises their ability to live for months rather than just weeks. So we'll talk a little bit about that. The winter bees. So this is a diagram of a model by some colleagues at Penn State University where they link these different conditions of how a colony kind of is triggered to start and is stimulated to start raising those winter bees. And it all, of course, starts with the outside conditions where the days are getting shorter and colder. There's fewer flowers out there for them to be foraging on, so that reduces the amount of foraging and the amount of food that's coming into the colony, obviously. And then that has a positive feedback loop, or I should say negative feedback loop on the amount of brood that is being reared, so they're naturally brooding down at this time. As you sure I'm all are well known, that the larvae produce its own pheromone that is a stimulant, especially for pollen foraging. So with fewer larvae within the colony, you're going to have even further reduced foraging. When you have a lot of unemployed foragers, these older aged bees that are just sitting around not doing anything, what that does is that starts to trigger an increase of a forager pheromone. So there's kind of like, a pheromone that the foragers themselves are putting out. When you have too much of that, then rather than those nurse bees graduating to become forager age bees, because there's this kind of backlog of foragers to begin with, you have this slowed behavioral maturation, so the nurse bees stick around and don't graduate to become foragers as early. So you have this kind of backlog in the division of labor within the colony. And then that excess number of nurse bees then triggers the colony to want to invest more in rearing these winter bees while they're in the larval stage. Does that make sense? So all of it really is kind of triggered. And so what happens when you have kind of this slow decline going into winter and then you have this really warm spell, it can often confuse the bees, right? But they don't have any control over it. But in general, that's the model of what they think is going on, of what causes colonies to start raising these long-lived winter bees. So these diatoneous bees, what are they? What makes them different, I think, from nurses or foragers? So this is, again, from that same group, a model of the green here is indicated, the larger that it is, this is what the nurse bees look like. The red indicates what the foragers look like, and the blue indicates more of what the winter bees look like. And so the nurse bees obviously have really big hypopharyngeal glands, right? The brood food glands that they're feeding the young, but the forager bees do not. The winter bees don't have active hypopharyngeal glands either. So they're not kind of long-lived nurses, they're different from nurses. The foragers have these high levels of juvenile hormone, whereas the other two do not. But what really makes the winter bees special is that they have this protein, this special protein that you've probably seen or read about in the beekeeping journals, vitellogenin. Vitellogenin in queens is actually the egg yolk precursor that she uses to nourish and to make all of those eggs grow and become really big and have lots of nutrients for the developing offspring. In winter bees, it's stored in their fat bodies, kind of the analogous of our liver within the abdomens of the bees. And so they just get loaded up with this, and then that kind of bleeds over so that they have high levels of just general protein within their bee blood as well. So they're just loaded up with kind of fat and protein stores. And so because they're not working themselves to death like the summer bees do, they can live very, very long. And so you have these kind of older bees that once spring actually, comes about, they'll be able to then activate their hyperpharyngeal glands with the stored protein that they have in their bodies and raise that first, cohort of brood so that the colony can get going into the spring. So again, depending on how long your winters last, having these winter bees within the colony with all of this stored protein within their bodies, not in the combs, is really, really critical. I said that going into winter is probably the most important predictor and indicator of success coming out of winter. And that is because these winter bees are so important that when they're being reared, they want to be free of parasitism or other stressors that will make them more likely to survive the winter. So things like Varroa parasitism is a huge stressor that causes a lack of vitellogenin within the winter bees' bodies because the Varroa actively feeds on the fat bodies, which is where vitellogenin is stored. And one thing that's really, really important, I understand you had Sam Ramsey down last year. Was it last year or 2 years ago? Something like that. This is his work, so I'm not going to spend a lot of time on this if you saw his talk on this. But this was a really insightful paper because it showed that the Varroa mites are feeding on the fat bodies of the bees, ergo weakening these winter bees. But more importantly, this is the thing that I absolutely love showing beginner hobbyists. When I say, what are the mite levels within your colony? You say, well, I don't have any because I don't see any. Well, what this work had shown is that he actually looked at all the bees and he found where the mites were actually hiding out on their phoretic stage, where they were hanging out on the body surface of the adult bees. And what he found is that for every one bee that you see on the top of her thorax, there's 49 that are underneath on the belly that you can't see. So you're only seeing 2% of the mites if you're just actively scanning the workers and seeing how many of them have a mite on their back. So this is why, and I think this came important yesterday, about monitoring. 25% of beekeepers in New Zealand do not monitor. That's great because it's more like 40% in the US, right? But monitoring is really, really critical because if you're just waiting and looking for the presence of the mites, it's already too late. They already are overrun with them. So you need to do your alcohol washes or some other means of assessing the number of mites within the colony, especially going into winter, especially in your, what would it be, April for you? March? Is that your kind of time frame when the mites are taking off and they're making the winter bees? Sorry, I'm trying to add 6 months in my head on the fly. I can't do it. But again, this is why. This is a really critical time when they're making those winter bees. So control them. You don't necessarily have to treat with synthetic pesticides or anything, but anything that knocks the mites down and controls them so that they're not going to be having these negative effects on the winter bees. All right, so you've already done that. You've controlled your mites. You have a huge proportion of winter bees within your colonies. That's great. Most other social insects, the colonies die and, next year's queens kind of fly off and go into some sort of hibernation called diapause and then start a new colony next year. Not honeybees, of course. They overwinter as a colony. They never really go dormant. They might go behaviorally dormant, but they don't ever kind of go dormant themselves. And so what they do is they cluster. So they retain their heat by getting closer and closer together. So they share their body warmth as they're kind of naturally rubbing elbows with each other, so to speak. And I put this degrees Celsius in here for you. I had to do the calculations, sorry. In the US, we're still on the Fahrenheit system. But this is the temperature by which the bees start to cluster, so actually still fairly high, but for the bees that like their hives at about 95 degrees Fahrenheit, which is 34 degrees or so Celsius, that's pretty cold to them. At about 10 degrees Celsius, that's when they stop foraging activity and they all remain within the hive. That cluster density, so they kind of get closer and closer to each other, continuing to increase sharing that heat and retaining that heat. That cluster, they can only get so close to each other, right? So that cluster gets at a density, maximum density, at about 10 degrees Celsius outside, at which point they then need to generate the heat, right? So they start doing kind of jumping jacks, right? They start exercising their wing motor muscles without flapping their wings, and that helps to generate heat that they can then share to others, and they can survive for long periods of time, retaining and even generating heat, even when it gets down into the minus 20s for prolonged periods of time. Now the cluster, I said before that what you want to do going into winter is you want to make sure the honey is above the cluster, so you start out with a cluster down here. The bees move both laterally and then upwards because of course heat rises, so it's easier for them to uncap the honey above the cluster during the middle of the day, get that food so they can continue to generate heat. So by the end of the winter, they're coming out with the cluster up there. This honey, any honey down here has all been eaten up, and the cluster's more up there, right? And the cluster itself has kind of a gradient of temperatures. So inside the cluster, especially if they're still rearing brood, which in some parts further south I'm sure they're not doing, but at least in where we are in North Carolina, they never really truly go broodless, or at least they always have a small little patch of brood in most places. And so they keep that at brood nest temperature, but the outside of that cluster, they can actually be fairly cool, and they serve as an insular blanket, so the inside can stay warm, and they're the ones that kind of get cold. Of course, they get sick of that, so they kind of burrow their way into the middle, pushing the ones in the middle outside, so there's this kind of constant fight to get to the inside where it's warm. But that's what happens. You have these kind of different gradients within the cluster. Now, what's interesting about this is that, when the colony is actively rearing brood or actively keeping the brood warm, this is when they're expending the most energy. So this y-axis here is the metabolic rate of the bees. And so when they're starting to cluster and they still have lots of brood, they're using a lot of energy because they have to heat that brood up to 34 degrees C, and that just uses a tremendous amount of energy. But as they decrease their brood rearing and they start to cluster and more about retaining heat rather than producing it, they're actually not expending as much energy as possible. When it gets really, really cold and they have to produce a lot more heat, of course, their energy expenditure goes up. So if you want to have your stored honey go the longest, last as long as possible, you actually want the bees to be without brood, but without having to generate a lot of heat. So that's around 10 degrees Celsius or so. Now, of course, we, at least maybe you do here, but at least in the US, we can't control the outside temperature very well. Yes, that was a joke, a bad one, obviously. But, this is why you actually want the bees to be in cluster and not to have these warm, sunny days every single day in the winter, because then they're going to be rearing brood and expending more energy. You want the bees to kind of be in that quiescent state where they're not using as much of their resources over a period of time. Now, a lot of these things, you can buy a FLIR camera, which is a infrared camera sensor. So if there's a representative of FLIR here, I'd be happy to take your bribe right now. But these are about $100 or so in the US. I have no idea what they would cost here, but they're very, very handy. These are some pictures from our yard which shows this is kind of later in the winter where a lot of the clusters are on the top, as you can see that. So the darker colors are cooler temperatures and the brighter colors are the warmer temperatures. But these pictures really aren't that great because it's really more showing the heat on the outside of the hives, not so much on the inside. But if you play around with these cameras a little bit and kind of tweak with the settings and things like that, you can come up with some much better pictures, things like this, that are much more resolute. So of these two colonies, which one do you think is dead and which one do you think has a nice active cluster? Right? Pretty obvious, right? That one just doesn't have any temperature gradient at all. That's a dead out. This one has a nice cluster. You don't want to be touching that, opening it up or anything like that. Just let them be. But for these dead outs, this is a really good way to go in before it warms up and they might start robbing each other out or those kind of things. You can remove these dead outs long before you might otherwise. And so having these infrared cameras are really, really good. Here's just some other examples of this. This is kind of two examples of a cluster. Here's one, isn't this interesting, where it's really spread out, elongated, and there's a lot of them up here, trying, to escape or something. I don't know what they're doing. This one's a lot more tight and focused, right? So you can kind of get an idea of the dynamics of what's going on inside your hives without having to crack the lid, because you don't want to be doing that, because then that, releases a lot of heat and it destroys the propolis seals that they've established and everything like that. So, having, I think, these cameras can be really, really helpful in the winter so that you're able to come out of the winter a lot stronger. Now, you can do, one thing that's true about beekeeping is that you can do everything right and still everything goes wrong, right? So a lot of times you go into these dead outs and you see things like this, where you just have this kind of small ball or cluster of bees, maybe getting moldy or so. They just kind of didn't have that critical mass to be able to retain or generate heat, and they just weren't able to make it. A lot of times you'll see this. How many of you have seen, going into a dead out and you see just the butts of the bees sticking out at you, right? This is very common of overwintering losses, because what these bees are doing, what they do when they generate their heat, they do that jumping jacks to create heat in the cluster. They actually stick their head in an empty comb cell, and they move their wing motor muscles, and then that heat radiates throughout the comb. So it's kind of like your nice warming floors that you have here, right, to just kind of permeate throughout the cluster, not from direct body contact, but by kind of warming the comb. So this is the last gasp of them doing that, but then they ran out of food or energy or something and died that way, which is, I don't know, it's not really a good way to go, in my opinion, butt faced up. But anyway. North Carolina, again, it's kind of the southern U.S. Where we don't have very long winters. We do not need to wrap our colonies, but I've spent time in upstate New York and other kind of cold places where it is much more common. I would imagine that, again, here, the further south you go, you may or may not wrap your hives, but is this even a thing here in New Zealand? Wrapping hives to retain heat? Not really? All the way, okay, so occasionally, okay. Yeah, for some reason, again, I think it comes from a lot of these textbooks that were written by kind of northern beekeepers or something. A lot of southern beekeepers think that they need to wrap their hives, but they really don't, just to have this kind of tar paper or something like that to help collect some of the solar energy, but it's really not necessary. As long as your colony has enough food, again, they won't freeze to death. All right, so let's talk a little bit about kind of spring management. Again, many of this is probably beginner stuff or stuff that you have a different take on, but so I'll just kind of go through it very quickly. But again, one of the successes of coming out of winter is going into winter properly, but then what to do when you're actually finally in spring and what to do. And And because we don't do this by the calendar, every year is different, every location is different, we have these kind of different early, middle, and late seasons, which I will define. Early spring is when it's still really kind of cold out. It's not beekeeping weather yet, but it's still something where you need to hopefully prompt and push the bees, maybe do a little triage to make sure that they have enough food so that they can make it out of winter officially. Mid-spring is when you're really looking at building up and swarm prevention. And then finally, late spring is when your main nectar flow is, or oftentimes the bees have kind of gotten through that major swarming period, and hopefully you're just kind of making your honey. So for that initial early spring inspection, you want to make these very, very quick, very short duration. You don't want them to be losing a lot of heat. You do want to be going into the colony when it's still fairly warm out, so they're not going to lose as much heat as if you go in when it's still freezing. But you want to be very quick and make a very quick assessment within seconds, not minutes. You want to look not for the queen herself, but the evidence of her. What kind of brood does she have? Do you have all stages of brood, or are you just seeing eggs? Or are you seeing eggs and young larvae only? That'll give you an idea of when a lot of that is going to hatch out and when the colony is going to start building up again. We do this in the US really quickly in about 2 or 3 seconds by doing a cluster count. We overwinter with 2 double deeps. You just split the double deeps. You count the number of frames that has bees on it. That's where the cluster is. And so you can have a very quick idea of the colony population. Doesn't tell you anything about the brood necessarily, but at least the population. If you're good at hefting and lifting, the hives, you don't really even have to go into them to get an idea of the honey stores. Honey, of course, is the heaviest thing within colonies. So if they're really light, if it's really easy to just pick them up with one hand, you might have to do some emergency feeding of some sugar syrup or a candy board or something like that to make sure that they have enough carbohydrate resources to be able to keep generating that heat, raise that first batch of brood, and come out of winter. Again, it's a good idea to remove all of those dead outs or any problem colonies so they don't get robbed out, they don't spread disease or any other things. It's also a good time to do a necropsy and an autopsy on your bees to see, why they might have died. Did they die not having enough food? Did they die of some sort of disease? Is there, a way to learn from potential mistakes or things going wrong? So this is a picture that I borrowed from Dewey Caron, a real hero of mine in the US, a longtime colleague at the University of Delaware. I was really disappointed to learn that this picture was staged. I thought it was just a natural thing of just a really bad beekeeper. But it was staged, and it was staged in a talk just like this of saying, is this a good behavior for a beekeeper for an early spring inspection? And the answer is no, but what several things give this away? Why is this bad behavior for an early spring inspection? What gives it away? He's sitting on the lid, which means he's taking his time, right? Which you don't want to do for an early inspection. What's he wearing? Yeah, if you're wearing a winter coat, you shouldn't be sitting on a lid just going through frame by frame, taking a kind of quick look and see how pretty the queen is, right? This is a bad kind of thing to do. You want to make sure that you're just in there, in and out really, really quick. You don't want to be taking your time. Thank you for this publication. I kind of forget when it was published. I think it's probably 20 years ago now. But this is something that we in the US use a lot, and it came from here in New Zealand. Oh, it's Australian, I'm sorry. Okay. Well, I knew it was a southern hemisphere somewhere. But this has been really, really great, and I skipped a lot of slides here for the sake of time, but this is something that, again, I think has been really, really helpful in both how to feed sugar, carbohydrate sources, as well as protein. I think, at least in the US, I don't know how it is here, but in the US, beekeepers tend to be really, really good about feeding a sugar syrup. In fact, they probably overfeed. They're constantly keeping the sugar syrup on, which to me might not be the best thing at certain times of year. Other times it's absolutely necessary, but not all the time necessarily. But what we're not really good at in the US is feeding protein. And so protein feeding is really critical, again, coming out of winter, so that those overwintered bees or those first flush of those nurse bees are able to regenerate their hypopharyngeal glands in their heads to start feeding brood. The more brood that they're rearing coming out of winter, the stronger and quicker they will build up, right? So this is a picture by Zach Wong, my colleague at Michigan State University. These are some hypopharyngeal glands, these grapey looking things that are not active, and then these are some hypopharyngeal glands that are just swollen with all of this brood food. So this is what you want to get at, and this is how, providing some supplementally fed protein can really boost that. For our operation, our time of year, we do this right after winter break, really early in January. They're just starting to brood up for us. It's still winter out, but they're starting to brood up. We put in some pollen patties, and that really kicks them into gear. And within about 2 or 3 months, we're having to prevent swarm control. Now, I make the distinction, as does the fat bees, skinny bees, between pollen supplement and substitute. Substitute are non-pollen, non-natural sources. They can be cheaper, readily available. There's all sorts of recipes out there, but in essence, it's not based on natural pollen. A supplement is mostly based on natural pollen that you can make into patties. The easiest and the best recipe that we have is 1 part corbicular pollen that we caught the previous spring out of our own colonies. You don't want to use somebody else's who you don't trust. To 1 part sucrose, table sugar, and then you drizzle in just a little bit of a thick sugar syrup so that it comes together the consistency of a cookie dough, and then you make your patties after that. And so we often trap our pollen in the late spring when we have a major pollen flow. Then we put it in the freezer. We kind of grind it up in a coffee grinder before we do so it's nice and powdery. And then in the winter, we're able to make these for free, in essence. We're able to make these pollen patties from our own pollen and throw them into the colonies. Another thing that we like to do is we like to make our own frames of bee bread. So pollen patties, supplementally provided pollen, does not get stored, right? So the nurse bees consume it and then they turn it into to brood, right? They put it directly into brood. They never store it. But research has shown that bee bread is way better than substitute or even supplement, and even better than pollen itself. Because bee bread is stored pollen, and that stored pollen is partially fermented. So there are these beneficial yeasts and other microbes that colonize the stored pollen. And it starts to break down the outer coating of the pollen grains so that it's more nutritious and more accessible to the larvae that are consuming it. And in fact, there was this one study, really interesting, that showed if you track the source of the protein from the bee bread, whether it be plant-based, the pollen itself, or the yeast-based, small microscopic animals, the larvae, the honeybee larvae, actually get more of their protein from the yeasts than they do the pollen. So what that means is that bee bread is more nutritious than even pollen itself. And so the supplements that we provide them, yes, is providing a lot of nutrition, but the best nutrition is the stored food. Has a lot of the lipids, vitamins, minerals from these kind of microbiota within the fermenting pollen. So what we do in order to make frames of bee bread is we take that same pulverized pollen that we collected from our node colonies the year before, we take some empty frames, and we drizzle the pollen into the empty frames, and then we spray a little sugar syrup on the surface to make it nice and tacky so that it doesn't fall out. We put them on both sides. And then we throw it into the middle of a strong colony, and then within about a day or so, they pack it all in. And so you just take it out after 24 hours, and it looks like this, an entire frame and nothing but bee bread. You do that in the middle of the summer when you have nothing else to do, because, there's always a lot of downtime in beekeeping. And you throw those into the freezer, right? So you can make your own pollen patties ahead of time and throw those in the freezer, or you can make your own frames of bee bread, throw those into the freezer. And then when you're coming out of winter and you really want them to brood up and they just don't have a lot of pollen coming in or anything else, you can throw this right in the middle or adjacent to the brood nest that they've started. They will turn this into brood like that. And it's a really great way to boost them really, really heavily in the spring. We do this mostly when we want to raise drones for mating with our queens. So making these frames of bee bread is another real neat trick. Okay, so that's enough of that early spring management. The mid-spring management, what does that entail? This is, when things are warmer out and you have a lot more time to go through, really assess, do a much better treatment, equalizing, right? You're able to manipulate your colonies a lot better. Uniting them is necessary. Finding kind of lagging queens and replacing those is really, really good. And then, of course, to stimulate brood nest expansion through reversing. So coming out of winter, you often have these empty brood boxes because the cluster is moving up. If the cluster wants to move up, you just reverse those brood boxes. So now you have the empty brood box above so that they will fill that, and that will help expand. It also helps to reduce swarming. And then in the late season, the late spring, hopefully you've done that, you've expanded the colony, they're rearing brood, but you want to get them as populous as possible without swarming. And this is the razor's edge that we all walk as beekeepers. And so that late spring management is where you want to have minimal disturbance. You want to put on your honey supers so that they can fill them. I can start getting beekeepers in fistfights in here, talking about whether or not to use queen excluders. And of course, this is when you don't want to be feeding any acaricides or any sort of medication in your colonies while they're obviously filling their honey supers. This is very standard for us. This is always what our bee yard looks like. I wish we all did. Actually, we're the worst example of a beekeeper operation because we do things for science. We give them disease on purpose, right? We let the mites build up. We take their queens away. We're just the opposite of a good beekeeping operation. So that's a good excuse of why our hives don't look like that, but I wish they did sometimes. But one thing that I want to hammer home here is this nonlinear relationship between a colony's population and honey yield. And again, most of you probably already know this, so forgive me. But this is why, when a colony is quite small, say it has 10,000 bees coming out of winter, their effort is to grow, right? Their effort is not to store honey, therefore to collect honey. So the proportion, which one is that? It's the left-hand side. The proportion of the forager-aged bees that are engaged in nectar foraging versus pollen foraging is very low. So only about 50% of the bees or so, when the colony is very small, is collecting nectar. The rest of them are collecting pollen so that they can make more brood, right? So as the colony population goes up, what happens is that percentage of nectar foragers goes up correspondingly. So when you have a colony of 60,000 bees, 90% of them are foraging for nectar because the brood nest can only get so big. The queen can only lay so many eggs. So once they get to a certain size, they don't need proportionally to collect as much pollen, and so those foragers are really collecting nectar. So when you have a really, really large colony, the vast majority of them are collecting nectar, meaning that they're storing a lot more honey. So what that means on the right axis, this is the kind of accumulation, this is your honey yield as a function of colony size. So the bigger the population, the exponentially increasing honey yield you're gonna get. Another way of putting that is that a colony of 60,000 bees is going to be collecting way more honey than the combined honey yield of 2 colonies of 30,000 each. Same number of bees, but you're gonna have way more honey produced in a larger colony than in 2 medium-sized colonies. Make sense? And this is why, again, because of the dynamics within the hive and the proportion of those forager bees collecting nectar. So again, one last thing that I'll touch on but won't dwell on is swarm management in the middle and late season. Now I know that none of your hives have ever swarmed, but for your neighbors, yes, this is important for them to understand. Now we want to have these really large colonies for honey production, but as I just said, the larger the colony, the more overpopulated they get, the more congested they get. That is the trigger for swarming. So that is their natural cue to want to reproduce at the colony level. Lack of queen pheromone and even brood pheromone circulating through the colony because they're just too congested in there, that's what triggers them to swarm. So of course, to minimize that, what you do is you want to add space. You want to decongest the brood nest so that they're not going to swarm. But it's, again, always a razor's edge that we often lose, unfortunately. So again, reversing, reorganizing, making splits. If you see capped queen cells, which means they're really on the road, they're just kind of dying to swarm on you. Doing that checkerboarding, is that the term that you use down here? Checkerboarding, where you're taking out frames of capped brood and you're putting in frames of foundation. So you're really knocking them back. So every other frame in the brood nest is now empty, and that really alleviates that congestion. You can then take those frames of capped brood, brush all the bees off, put them into other kind of lagging colonies to give them a boost when they flush out. So there's lots of things that you can do to have your cake and eat it too, right? Come out of winter, really strong, populous colonies that hopefully don't wind up in the trees. I'm a real advocate, of course, of every beekeeper harnessing the power of making your own queens so that at this time of year, you're able to make your own splits. Why have them swarm when you can just capture them and split them in half, and now you've retained all of the bees? And the only way that you can really do that and keep that momentum going without them losing about 3 to 4 weeks of raising a new queen, there's nothing wrong with that, but it does kind of delay things in that kind of springtime. You can make the splits yourself, and you can either make them equal so that both halves of your splits are the same size, same population, same distribution of brood and honey and food and everything else, or you can make them unequal so that you have one that's not backed a little bit, and that's your main honey producer, and then you have another split that is able to kind of grow up, alleviate the congestion from your main unit, but then you're able to retain and grow that other unit as well. We have this thing in North Carolina we call clean and dirty splits, or I should say clean and dirty splits, where if you have a double deep and you don't have the time or the wherewithal to want to go in and find where the queen is, what you can do is you're going to split the 2 brood boxes apart, go in 3 days later and see which one has eggs, that's the one that has the queen. And then you put the new queen in the one that doesn't have the queen. Now, of course, that's dirty split in the sense that you don't have an equal proportion of the different resources and everything within each unit, but you can do a lot really, really quickly if you need to knock them back fast. Okay, and so with that, again, hopefully that wasn't too basic or too introductory, but hopefully you had some new things that you've learned or a different perspective from us up north, and thank you so much for your attention. Yeah, I was told that we have time for just one or two questions if anybody has any. Please use a microphone, or otherwise I can repeat your question. Yes, sir.
Yes, David, interesting talk. I was happy to see that you'd referenced the book Fat Bees, Skinny Bees, which was written by an Australian, I forget his name. But one of the problems they have over there is particularly their autumn pollen sources are quite low in lipids. And A, I'd like to know if this is the same in the States, and B, if you've done any research around lipid profiles, and particularly supplements or autumn profiles to build up your bees for the winter.
Yeah, great question. And I guess I'm going to use the scientific weasel out of that and saying, well, it depends, right? So that's going to be my answer to that. The different pollens in different locations are going to vary tremendously, right? But in general, I think a lot of large-scale commercial operations have come up with their own either supplement or substitute that they use and swear by. It works for them. And a lot of times they do add lipids or other minerals or micronutrients in addition to the macronutrients. The problem is that they haven't been side-by-side tested blindly empirically. So I think as an industry in the US, we're quite behind in our nutritional research at the colony level. And a lot of that is because the bees are still foraging as they're doing that. So it's really hard to control the other forage of what's coming in. So you need really huge sample sizes. And a lot of the large-scale commercial beekeepers, they don't want to do a don't feed treatment that they're not going to build up at all. So there's some challenges with that. But I think you're right, that there are a lot of opportunities to be able to do that, but it varies from operation to operation. And it's just kind of like, it's just like babies, right? There's only one cutest baby in the world and every mother has it, right? There's one best kind of pollen patty recipe and every beekeeper knows it and is unwilling to share it, right? So at least that's how it is in the US. We have a lot to do on that. Yes, sir.
David, I start my question by apologising. I want to ask you about something you didn't talk about at all.
Perfect.
But an issue that you know, if you heard Oksana talk yesterday, that happened a number of years ago, and I think still happens, is Nosema's in early spring, and what we call rapid spring dwindling disease.
Yeah.
And I use your work as a sort of Bible on the lifespan of bees when they've both got Nosema ceranae and Apis. Just wondering if you can make some comments about Nosema in early spring in the US.
Yeah, Veromorpha Apis. Yes, that's right. I'm always catching myself as well. Veromorpha, Apis, and Cerana. We have a lot more work to do on that. A lot of the textbook, a lot of our mentality is still thinking about Veromorpha apis, where it's kind of a winter disease, a confinement disease, fumagillin and other kind of antibiotics cleanse out the gut and is really effective on that. Veromorpha cerana is not that. It responds sometimes to antibiotics, other times not. And so whether or not it's even effective is very unclear. A lot of times it clears up on its own. It happens in the middle of the summer, not just in the winter. So there's a lot of seasonality to it that we don't understand. And my guess is a lot of it comes down to the nutrition and the immune system capacity of the bees. And so if they have stressors and other things from nutrition, those things can flare up and then go back down again. So I think it's a lot more squirrely. But we don't have the same kind of spring dwindling that was described by Okasa and others. So I'm not really sure how to address that. It's not as systemic as in our neck of the woods as it is here. So. Well, again, thank you very much. You guys have been great. Fantastic.